Now students, we will discuss secondary structures of the proteins. So, we have already discussed whenever two amino acids unite to one another, there is formation of a bond, we call it a peptide bond. So, during the formation of this peptide bond, there is release of a water molecule. So, what happens? There is also involvement of a chain. There is also involvement of polypeptide backbone. Now, what is this polypeptide backbone? So, this is the structure of the resultant unitedness of the two amino acid. So, with the help of this figure, we will find out that what is a polypeptide backbone. Polypeptide backbones is actually the repeating sequence of N, C, C, N, C, C in the peptide bond. So this repeating sequence is actually making a structure. This is making a backbone to which we are calling as polypeptide backbone. However, what happens that the side chains, these side chains or the R group is not the part of the backbone. Now, we will discuss the structure of secondary amino acid on the basis of these polypeptide backbones. So our ultimate purpose of this video is to understand that what is the secondary structure protein. So before going into detail of the secondary structure of the proteins, we discussed that what is a polypeptide backbone, because in our text, in our literature, we are repeatedly using a term that is polypeptide backbone. So now we are clear that this repeating sequence of N, C, C, N, C, C is actually a peptide backbone, polypeptide backbone. Here again, in this N, C, C, N, C, C is a backbone. Then again, there is repetition of this backbone. So this repetition is basically making polypeptide backbone. Now, these polypeptide backbones does not assume a random three-dimensional structure. Actually, what happens in the secondary structure of the proteins, there is a regular arrangement of amino acids near each other in close proximity. So this is also a linear structure. But what happens that in the case of the secondary structure of the amino acid, these amino acids are near to one another. There is involvement of the alpha helix, there is involvement of beta sheets, there is involvement of beta bands. So what was in primary structure, there was just a linear structure of the amino acids. But in secondary structure, we are actually discussing helix. We are discussing about the sheet. We are discussing about the band. We are actually discussing about the alpha helix, beta sheet, and the beta term. We are discussing that in the case of the secondary amino acids or the secondary proteins, the amino acids are actually in close proximity to one another. In this case, we can find the alpha helix, we can find the beta sheet, we can find certain turns to which we are calling the beta turns. So this figure is representing a secondary structure of the amino acid. So what this figure is representing, this is representing that this helix, which is actually the alpha helix, is stabilized by a hydrogen bond between the atoms of the polypeptide backbone. So these dotted lines in this structure are actually representing the hydrogen bonds which are present in the secondary structure proteins. So now going into detail of the alpha helix, that what is alpha helix? So this is actually a right-handed spiral structure. This spiral structure is the alpha helix. The side chains are actually extended outwards of this alpha helix. Now, this helix is also stabilized by the hydrogen bonding. So, these dotted lines are representing hydrogen bonding, which are between the carbonyl oxygen and the amide hydrogen. So, in each turn of this helix, this figure is representing that this is a turn. So in each turn, there are 3.6 amino acids. So alpha helical segments are found in many globular proteins like myoglobin, troponin C. So what is the total length of this helix? The length is the 12 residues, approximately 12 residues or the 12 amino acids. So, and 3 helical turns. So this is actually representing a one turn. So these 3 helical turns are making the right-handed structure of the alpha helix. So almost 12 amino acids are making a alpha helix. This is again representing that this is a hydrogen bond between the carbonyl oxygen and the amide hydrogen. This again figure is representing the spiral structure of the amino acids or the secondary structure proteins which are found in the hairs, nails and bones. So we have already discussed that alpha helix is a right-handed structure. It is a spiral structure. It is tightly packed. It is really, really tightly packed in the coiled polypeptide backbone. So, but what is happening that in this case, these side chains are actually extending outward. Why these are extending outward? Actually to avoid the interfering sterically with each other. It is actually extending outward to maintain the shape of the protein. So, where these secondary structures are found? These are found in our hairs, in our skins, they are found in keratin. So, keratin is entirely alpha helix. These secondary proteins are found in our muscles, for example, in the myoglobin. Myoglobin is also a alpha helical structure. So, this is all about the alpha helix, that in alpha helix, the basic thing is that we are getting hydrogen bond, and this hydrogen bond is between the carbonyl oxygen and the amide hydrogen. So this hydrogen bond is actually stabilizing the structure of the alpha helix. This hydrogen bond is actually stabilizing the structure of the secondary protein.